In this video, we will discuss about CAR-T adoption to a hostile tumor microenvironment. In the previous video, we have understood how we can improve the anti-tumor efficacy of CAR-T. So we will go in the same directions and try to do further more modifications so that the CAR-T cells become hostile. CAR-T cell uh, get themselves adaptable to the hostile tumor microenvironment. So some of the changes that are commonly known is the knocking down the immune checkpoint inhibitors. So there are two categories of immune checkpoint inhibitors. First one is this intracellular immune checkpoint inhibitors such as LCK PTPN2, SHP1, and also the surface checkpoint inhibitors, which are more commonly known as immune checkpoint blockade therapies, that is by knocking down the PD1 and CTLA4. This will enhance the CAR-T cells adaptation towards tumor microenvironment by reducing the tumor T cell suppressions from the tumor microenvironment factors. Other things that can be targeted is this avoid fratricides, or the genes that help in these fratricides can be downregulated. This fratricide actually means that when the one immune cells is trying to kill the other immune cells. So for example, the receptors on the one immune cells can detect antigens on the other T cells. It might CAR antigens on the other T cells. So then it might trigger the killing of the one immune cells by the other immune cells. So these things can be avoided by downregulating these fratricide genes that take place in this process. And the metabolic enzymes that we have discussed extensively in the previous video, and also when we are discussing the previous paper, that is, for example, by downregulating this TGF kinase, it will improve the T cells efficacies, and will improve the T cells efficacies, T cells anti-tumor efficacies, and also the T cells half-life time. And the other things that can be targeted is by inhibiting the receptors that suppress the T cells activations. For example, this TGF-β and adenosine, it plays an inhibitory role, that is by suppressing the activated T cells. So these things can also be inhibited to further improve the CAR-T cells adoption to TME. And the dead cell program. So one of the common ways this tumor microenvironment behaves is by triggering the dead cells into the immune cells by interacting with the dead cell receptors of the immune cells. For example, the fast receptors. So if these fast receptors can be knocked down, then tumor microenvironment will not be able to trigger dead cells using the fast receptors. And the other things that can be changed is to make the gene regulations that will cause the epigenetic changes in a way that the genes that helps in the anti-tumor response could be upregulated, whereas the genes that helps in the tumor progressions, or that gives the negative feedback to the CAR-T cells, will be suppressed. So these are some of the changes, or the main categories of the changes that are discussed in this figure. So if these things can be adapted into the CAR-T cells or engineered into the CAR-T cells, then it might increase the CAR-T cells adoptions to the tumor microenvironment, and thereby increasing the efficacies of the CAR-T therapies against solid tumors. Right now, one of the major concerns of this CAR-Ts is that they are not able to target the solid tumors with good efficacies. So by making these adoptions into the CAR-T cells, and making these changes into the CAR-T cells, will make it more suitable to the solid tumors. So, and right now the CAR-T cells are good for the tumors like blood cancers, but they're not good for the solid tumors because of its low efficacies. So I hope you understood some of the changes that can be done in these CAR-Ts to improve their adaptations to the tumor microenvironment. So this figure focus on the engineered part of the CAR-T cells, whereas previously we have discussed how we can modify the CAR receptors, that is chimeric antigen receptors, to improve the activations of the CAR-T cells whenever they interact with the antigens. So that's all for this video. If you need to understand more about this topic in detail, you can go through the reference that has been provided in the description. Thank you for your time.